Hi guys, what we're going to do is we're going to go through the NEAP topic test 1 today. So hopefully before we do go through it, because I'm going to go through the answers, you've already given yourself the 45-minute time period that's recommended. Hopefully you had a look as well on the front page. It says that it's 40 marks. Now, what I would usually recommend is that you do the 45 minutes. However, you give yourself a 5-minute reading time and then you give yourself 40 minutes to actually go through, so you have a mark per minute. Okay, so let's get started. Let's get started on this first question here. Okay, so whenever you come across diagrams, it's really important that you annotate them and you understand what they're saying to you before you even have a look at the answers, especially in multiple choice. So you can see that we have 2 reactants and then we have 1 product. So we can see that it's getting bigger. Okay. Hopefully with the Essendon scandal, the one positive thing that you took out of that is that they were taking anabolic steroids to make them bigger. So this is an anabolic reaction. So these are the sort of prompts that I want you to start to see when you start to look at this type of reaction. And we know that it's a reaction because there's an arrow. Okay. Now, in this we can see that hopefully you recognize that these are 2 carbohydrate molecules because it has hydrogen, oxygen, and carbon, so it's got the CHO. Okay, we know that these are monosaccharides. Let's see if I can write just mono up here. Okay, and now they're binding together to cause a disaccharide. So remember, mono meaning 1 and di meaning 2. Okay, so there's some sort of bond that's happening there. If you have a look at the first option, it says to you the formation of a dipeptide. So di is correct, but it's definitely not a peptide because this is a carbohydrate. Okay, just write it's a carb. So it can't be A. All right. Z is a monomer. Sorry. So a monomer, remember. So mono singular is a subunit. Okay. So Z is made up of 2 subunits. So Z itself is not a monomer. It's a disaccharide now. Okay, it's made of 2 monomers. Water can be an input to this reaction. So I'm going to go back to the formation of all biomacromolecules. It's a process called polymerization. Spell this wrong because I'm trying to concentrate on 2 things, all right? Polymerization, condensation. Okay, now I know you've all heard the word condensation because your parents have probably told you off about not putting the fan on in the shower because the water all condensates on the roof and the walls and can create some real damage in there. So that's the formation of water. Okay, and then your polymerization, you've seen the word polymer, so it's making a bigger molecule with many subunits. Okay, so that's what polymerization means. So at the end of this reaction, you are going to have water as a byproduct. Okay, so it's going to be an output or a byproduct. Same thing, different words. Okay, so in fact, when it's saying an input is incorrect because it is an output. So we can say that C is incorrect. So condensation is represented so that we know that that is correct because this is a polymerization condensation reaction because we are building up, we're getting bigger because we are making, okay. So let's move on to question 2. So hopefully this question is fairly easy. So when we go back and we have a look at that table from that Year 11 textbook, you'll be able to see in the first column, well, one of the first columns, that it goes through the monomers. All right. So remember, monomer is the subunits. They're the building blocks. Okay, so they're the units that are repeated over and over and over again, except for the lipid, which remember, a lipid is not a polymer. It's really important that you remember. A lot of students get this confused or they forget. Lipid is not a polymer. All right, so it says here monomer and polymer. So even if it did have and it does for the first section there. So it says A, fatty acids and glycerol, which are the two subunits or monomers that make up a lipid. But remember, A cannot be correct because a lipid is not a polymer. So we can get rid of A straight away. So my little rant worked. Glucose, that is a monomer, so we could say that is correct. However, glucose will go on to make a polysaccharide. I don't know if you can read that, it's very tiny. Polysaccharide, not a protein, okay? The monomers, or the subunits that do make up a polypeptide or a protein, are in fact amino acids, okay? So we can see that that's there at C. So that's correct. However, when you do your multiple choice, make sure you do read all the options. That's really important because there could be trick questions. So if we go look at D, that does look a little bit tricky. So nucleic acid and DNA. So they seem to go hand in hand. They seem to be all right. But the monomer of a nucleic acid, so the polymer here is the nucleic acid. There are two different types. You have your DNA and your RNA. So I'll just write that over here. So nucleic acid, there's two different types. So they're two different branches. So you've got your DNA and then you've got your RNA, okay? These are still the level of the polymer. All right, so you need to know what's going to make up your DNA and your RNA. Let's get rid of that so I've got more space. So in fact, what the monomer is a nucleic of the nucleic acid is your nucleotide. Okay. So we've got that there. So then we've got our nitrogenous base. We've got our pentose sugar. Remember, ends in OSE. And then we've got our phosphate group over here. All right, it's very important that you can draw that diagram. So the answer there, if we go back, is C. All right. So number 3. Let's see if I can get rid of this because I don't know if I have enough space. All right, so examine the following structure. So this might be a little bit distracting while I'm getting rid of these bits and pieces. So remember, when you come across a diagram, please make sure that you are annotating it. All right, so let's zoom in here. So let's have a look. Examine the following structure. So we have an R group. Okay, so this should start to ring some bells. All right, we've got an R group, we've got a carbon in the middle, got another group over here, and another one over here, and then a free floating hydrogen with your carbon. This R group, I'm going to give you a hint here, is the functional group. Ooh, don't want to do that. Functional group of the monomer and even the polymers of this. Okay, this one here is your carboxyl group. And this one here is your AIM group. Now, if you're doing chemistry, this will probably ring a few bells for you. This was asked on the 2014 biology exam, so you were asked to draw the molecular structure of two of the monomers that were found in a plasma membrane. And so one of them was a lipid and the other one was protein. So this is, in fact, the molecular structure of an amino acid. Okay, so every amino acid will have a carboxyl group, an amine group, and it will have its own R group, but this is individual. All right, so this is what makes one amino acid different to another amino acid. And remember that we do have 20 amino acids. So the way that in which they differ is this R group. Okay, so the correct answer for that is obviously B. All right, so moving along to number 4. Before I do that though, actually, I just wanted to go over the reason why it's not 3 was not a dipeptide, is because this is only a single amino acid. If it was joined to a another amino acid, then in fact it would be a dipeptide, and it would bind with the carboxyl group. So it would bind to another one around here, and remember that would then give us an output of water because it's a polymerization condensation reaction. So whenever we are given water, it's condensation. Can anyone guess what the opposite of that is? So when a reaction requires water, it's actually called a hydrolysis reaction because you need to hydrate. Okay, it's funny how these words that you use every day come into science. So just to clarify that with that. So question 4, an enzyme's protein structure. So sometimes it won't even say protein, it might just say an enzyme structure, and it's important that, and I'm sure you do by now, that you recognize that an enzyme is in fact a protein. So we'll go through for that. So remember, there's a different structures of proteins, in proteins, sorry. So transcription and translation is extremely important. We do go through it more in Unit 4, but I think it's important that you know about it and are aware of it in Unit 3. So that's the big picture that I have on the back wall with the funny green looking things. So it's making the proteins at the ribosomes essentially. So sorry, we'll go back to the question, I'm getting off track. An enzyme's protein structure is A, primary. So remember, the primary structure of any protein is in fact the order of amino acids. Okay, the secondary structure. Now I did mention about the R group and how it gives it different characteristics at the monomer and the polymer level. That's because it gives them the different characteristics and the individualism because they either repel or attract to one another. And that's where we see the different structures of the beta pleated sheets. And I'm going to make sure I write these because, keep doing that, pleated sheets. The examiners want full names, so it's really important that I beat on about this, okay? And then your alpha helix coils. Really essential that you know those back to front. Okay, so remember, it's like if you're all holding hands, and you have to hold hands because you're binded to each other because you're binded through that polymerization condensation reaction, but you don't like the person that you're standing next to. All right, so that's your R group, that's your individualism. You don't like the people that you're next to, so you want to repel them, so you're going to move backwards. Okay, and then the people next to you want to get away from you as well, so they're going to move forwards. So therefore you're almost going to have, if this is you, and then I'll draw the people that you're holding hands to, you're going to want to move this way and they want to move that way. So you're going to start to see this beta pleated sheet movement. Okay, and then the coils, there's obviously the coils like that. All right, but that's what gives it its flexibility, okay, or its functionality as well. So it's really important that you know that. Now, an enzyme, it can't be a functional enzyme if it finishes at that secondary stage. It must make sure it has a specific molecular structure. So it has to have a tertiary level. It's really important, I'm going to try and write this in black, it's important that you know that the tertiary structure is a 3D shape. Sorry, alpha helix coil. So it's a 3D shape. It's the function of that protein. All right, so it must have that in order to function. That's really important that you know that. All right, remember, it does require some chaperone proteins in order to get to that tertiary level. So you might get asked a question in an exam that says to you, if we take the protein and we work backwards to find its DNA sequence and then try and translate it, why can't we do that successfully? It's because you don't have the genetic code for those chaperone proteins to fold it into its tertiary structure, so therefore it doesn't work. So that's quite important to know. I might just write, running out of space again, chaperone proteins. Okay. So not all enzymes will need a quaternary structure. So the one that quaternary structure is when it has two or more polypeptide chains. Please make sure you write polypeptide chains, not proteins, because a protein is implying that it is a finished unit. So it must be a polypeptide chain, not a protein in this definition. So I'd write that into your prompt sheet. So quaternary structure is two or more polypeptide chains that are put together in order to give the protein, or in this case the enzyme structure. However, not all enzymes need to have a quaternary structure, so therefore the answer for this is C. A very important example of a quaternary structure in the human body is the hemoglobin that we see in our red blood cells. I would take the time to know that because it seems to pop up as general knowledge in the exams. Beautiful. So let's move on to question 5. Now I think this one is quite easy. So nucleic acid, so remember, let's see if I can move it so we've got a bit more space. That's better. So nucleic acid, remember we've got the two branches, so we've got DNA and we have RNA. All right, so they're at the polymer level. Now it's saying are only found in eukaryotic cells, so remember they're complex cells, are only found in the nucleus of a cell, found in eukaryotic cells and viruses, not found in viruses or prions. So before we go into that, you kind of need to know what a eukaryotic cell, a prokaryotic cell is, a virus, and a prion. Now we'll look more at viruses and prions when we come to chapter 5 and 6, which is the immune system, but viruses pretty much are non-cellular. I keep doing this, don't I? Non-cellular, okay? So they are not made up of a membrane. They in fact have a protein coat around some sort of genetic material, and that genetic material would be in fact a nucleic acid, okay? So you can have in a virus, you can have either DNA or RNA, okay? But it doesn't have a cell because it's coated with proteins, okay? A prion is actually mad cow disease, so it's an infectious, misformed protein that is almost contagious. So when it comes into contact with a normal protein, it changes its shape. So that's also non-cellular. So I'll put a little non-cellular box here. But this has genetic material, whereas a prion is just a protein. And so you know that that's only made up of amino acids. So in fact, that does not have any nucleic acid. All right. Your prokaryotic and your eukaryotic, the only difference between that is that your eukaryotic is more complex and it has organelles. Okay. They still both have genetic material. So if we go back to the question, sorry, this is really all over the place. So nucleic acid only found in eukaryotic cells. Well, we know that it's found in eukaryotic cells, prokaryotic cells, and viruses, but it's not found in prions. So we can get rid of A. Only found in the nucleus of a cell. While viruses, I've just told you, don't have any membrane, so they don't have a nucleus. Prokaryotic cells don't have a membrane because they don't have organelles, but eukaryotic cells do. But we know that all three of those have genetic material, so we can say that that's incorrect. Okay. Then we have a look at C. So it says found in eukaryotic cells and viruses. It is found in eukaryotic cells and it is found in viruses. So it doesn't say that it's not found in prokaryotic cells, so we can leave C as an open, but remember, always go through every option. So then we go to D, and D says not found in viruses or prions. The thing that's incorrect about D is it is found in viruses. Okay, so D is incorrect, and we can go back, and the answer is C. Okay. So let's move on to question 6. Oh, before I do that, it's really important, and I should have mentioned it before. When we talk about enzymes, I'm jumping back to question 4. I know we're on to question 6. When we talk about enzymes, and I'm sure that you were like gritting your teeth and yelling at me and telling me that I forgot to talk about this active site. Whenever you talk about enzymes, you must talk about their active site, because you must talk about their function with I'm hoping that you're getting the point of the importance of this because I'm going backwards and seeing how big my letters are. Structure and function. You must talk about them together. So when we have our upcoming enzyme sac, please make sure that you are talking about the different protein structures that help to give it the active site, which allow the enzyme to go through and do its function, which is to bind with the substrate to create product. Okay, really, really important. Okay, I think I might actually just clear all of this. All right, let's move on to question 6. So, studies of the organic compounds. So that should ring a bell as to we're looking at carbs, proteins, lipids, and nucleic acid. Found in cells have shown that lipids are made of chains of glycogen and fatty acids. Okay. B. Proteins are made of only carbon, hydrogen, oxygen. What is the big main thing that we're missing there? Good, I can hear you all yelling down the computer. Nitrogen. Okay. C. Proteins in plasma membranes are quaternary structures. Okay. Most of the time, and I think this is a bit of a strange question, I don't think you really get asked this question, but you never know. They are generally just secondary structures. So most of the time when you do have a look, if you go and pause and go Google this now, if you have a look at plasma membranes within protein channels, within the plasma membrane, you will have a look and you will be able to recognize the beta pleated sheets and the alpha helix coils and the random coils. So again, that's just a secondary structure. So most of the time they're secondary, very rare that you would see at all quaternary structure. Okay, the monomer of DNA is a nucleotide, and we know that we talked about that before, so that's correct. Okay. So sorry, going back to A, I don't think I clarified that we know that lipids are made of a glycerol molecule and 3 fatty acids. Glycogen is actually, hopefully this is a bit of revision from year 11, is a complex carb or a polysaccharide that we see in humans, which is used for storage. So just like you see in plants, you see starch, and remember it's a huge molecule. In humans, when we look at storage, we store our excess glucose as cool. All right, so number 7. So I think 7 and 8 is just going over organelles. So this should be fairly simple. So let's have a look. The organelle which would be expected to be more abundant, so have more in pancreatic cells, secreting insulin than in a bone cell would be. So I want you to think about insulin and think about what it is. Hopefully it's ringing a bell as to diabetes, some sort of chemical messenger. I'm not sure whether you know it or not, but it is a protein that will come into play a little bit later when we look at the types of signaling molecules, whether they're hydrophobic or hydrophilic. Hopefully those words are ringing a bell. So you, hydrophobic or hydrophilic or polar or non-polar means that they can either cross the membrane or they can't. So hopefully you know by now that protein is too big and it actually can't cross the membrane. So when we do look at insulin later, we know that that's a so there's 2 different types of chemical signaling molecules. I'm getting off track again, but I'm just going to go over here and write it for you. So when we look at molecules, there's 2 different types. You've got peptide versus a lipid or steroid. Okay, and the protein is the peptide hormone and the steroid is the lipid hormone. So lipids are obviously non-polar and your peptide are obviously polar. Okay, and your peptide is obviously hydrophilic, loves water, and your steroid is hydrophobic. So that's really important to know, because these ones here, your steroid, will be able to transfer straight into the membrane and can have a intracellular receptor, whereas your peptide hormones need to have an extracellular receptor. So when we look at signal transduction, I know that probably might not mean too much to you right now, but it's important for you to know later on. So that's important. So insulin is protein, so the receptor needs to be on the outside. Getting off a tangent. Anyway, so let's read the question again. So the organelle, which would be expected to be more abundant in a pancreatic cell secreting insulin, which is protein. So we need to make protein, we need to excrete it out of the cell. So what is responsible for making proteins? So let's have a look, and as we go through this, I'd like to write down the role for each of these. So your centrioles, hopefully you know that they look something like, need a pen for that. Oh, that did work. Centrioles look something like this, and they help with cell division. Okay. And to be honest, you'll generally only ever have two, so there's no variation in numbers within the centrioles. Your lysosomes are important in the destruction or the process that we will look at, which is apoptosis, which is cell death. So I'll write that down there. That's pretty important. We have a look at that again in when we're looking at signal transduction. So make sure that you're familiar with that. That's programmed cell death. I'm just going to write that there for you. Program cell death. So you generally won't have any inflammation when you have programmed cell death. It's a nice, neat process that your body has come up, literally a neat process. And then your ribosomes, hopefully you know that this is protein synthesis. Okay. And now, depending on where the protein is located in the cell, if it is on the ribosome, then that's going to be an extracellular protein. What am I saying? If the ribosome is, it's always going to be on a ribosome. If the ribosome is on the endoplasmic reticulum, it's going to be an extracellular protein, so it's needed by a cell outside of the one that's being made in. If it's going to be made on a ribosome that's free floating, that's for the cell. So it's not going to be needed to be transported by the endoplasmic reticulum, the Golgi apparatus. Okay, and then the last one here, we've got the nucleus, which is obviously the brain of the cell, and there's no variation with the abundance, because remember that's what. So let's have a look at question 8. So the organelles which contain the product of biosynthesis prior to excretion are. So this is really having a look at protein synthesis again. So because bio meaning life, and to be honest, proteins are extremely important because we already know that enzymes are a type of proteins, and to be honest, a lot of the very extremely important processes, pretty much all of the processes in our body, are done by enzymes, which are proteins. So proteins are really important. So biosynthesis, I would almost always say that that's probably talking about protein synthesis. And synthesis just means to create. Okay, so the organelles which contain the product, so being a protein, that's not how you spell protein, whatever, prior, so before it's excreted out of the cell. So we're obviously looking at a protein, an extracellular protein, that the mRNA was going to a ribosome that was on the endoplasmic reticulum. That ribosome then translated it into a protein which moved through the endoplasmic reticulum to the Golgi apparatus, which then put it into a vesicle, which then made its way to the plasma membrane for exocytosis. So as we go through this, I just want to go through the role of all of the organelles within protein synthesis. And I think I want to change color, so let's go with a pink color. Okay, so your ribosome, that's the site of transcription. And that just means turning your mRNA into protein. Okay, so remember there's 3 different types of RNA. There's mRNA, which is your messenger RNA. It's the only type of, well, no, it's not the only type, but it goes from your DNA to mRNA and takes the message from that outside of the nucleus, because your DNA cannot leave the nucleus. Your rRNA, which is your ribosomal RNA, pretty straightforward, is in the ribosomes, and then your tRNA, which is your transfer RNA, which comes into play later on with translation. So the ribosome, where proteins are made, pretty much your transcription. I'll change color with this one. Let's go for the pinky color. Okay, endoplasmic reticulum. As I mentioned before, the ribosome will be on the endoplasmic reticulum because it needs to go to the Golgi apparatus. All right, so A and B are pretty much your synthesis of protein. Okay, so it's the location. The Golgi apparatus is almost like the post office. Okay, puts a stamp on it, puts it into its vesicle and says, you're going to this destination. Have a good trip. All right. Your vesicle is the membrane around it. So it's the packaging that the post office gives to it to take it to it. So if we reread the question again, it says the organelle which contains the product, so the product being the protein of biosynthesis of protein synthesis prior to secretion. So before it, an excretion just means before it leaves the cell. And if we look at this process, it starts at the ribosome, which is on the endoplasmic reticulum, goes to the Golgi, and then the last organelle that it's at before it reaches the cell membrane is the vesicle. And that's what the question is asking for. Okay, prior to secretion. So the correct answer for that. All right. I'm just tricking you. I meant to write translation. Now, it is a bit off topic, but it's okay. You can, if you really don't want to listen to it, you don't have to. Oh, ran out of room. Okay, I'm going to come down here. So, it's really important that you know about transcription. So you're going to write the script first, and then once it's a hit, then you can translate it to other languages. So then that's translation. Okay, this transcription, that happens inside the nucleus because it's the DNA being turned into mRNA, and we won't learn about that too much until we actually look at DNA in a bit more detail in Unit 4. But translation, so remember that's turning the mRNA into protein. And this is out in the cytoplasm, which can be either on a free-floating ribosome or on one that's on an endoplasmic reticulum. But this always happens at a ribosome. Okay, and so remember the order that it comes out of, the protein, so these are all the amino acids, that's the primary. Okay, then remember the secondary is due to the interaction with the R group. Running out of space again. And then the tertiary is to do with your chaperone proteins. And then hopefully remember the quaternary. Can you remember what the definition is? It's 2 or more, did you remember it? Polypeptide chains. All right. Which one is its functional group? You've got a whole choice of colour here. The functional group is the tertiary, okay? So it's really important that you remember 3D shape. Yellow is really hard to read, and it's the function. And when you talk about enzymes, what is it you need to talk about? Nope. Active site. Okay, now that I've gone off my little tangent. Okay, we can go back now. Question 9. So this is to do with solutions. I would highly recommend that when you do come to solutions, I find it helps me when I draw beakers. Simplifies everything for me. All right, so examine the following cell. So it looks to me like you have a plant cell because we've got a cell wall. Instantly, that sort of thing should come to your mind. So no matter how much movement of water is going in or out, it's not going to burst. Okay? Really important. Sometimes they like to compare red blood cells and a typical plant cell. And the really key difference there is that one's got a cell wall and one doesn't. One will burst if too much water moves in, and one will not. Really important you know the difference. Okay, if the above cell was placed in a hypertonic solution, so I remember it's got an R in it, and I know this is a bit of an abstract way, however you remember, R means that it shrinks. That's for me, because shrink has an R. So therefore, if I draw, or even if I just draw my cell, it's going to shrink because water is going to be moving out. If water is going to be moving out, that means that there must be a higher concentration of the solute here and a lower concentration of the solute. All right, please make sure you are specific to solute concentration or water solute. All right, because this is water that's moving out. All right, and this process when water is moving, not the solute, is osmosis. And the reason why it's the water moving and not the solute is because it'll be too big or non-permeable. All right, so if the above cell was placed in a hypertonic solution, so remember water must be moving out, it would be expected that water would move out of the cell. So far so good. Okay, water would move out of the cell from a region of high solute concentration to a region of low solute concentration. So to here, where are we? So water would move out of the cell from a region, yeah, that sounds fine, of high solute concentration. Remember, we've got a low solute concentration. Okay, so we can get rid of A because it's not talking about the right substance for us. The cell would swell and become turgid. If water was to move in, then definitely we would describe it as turgid, and that's very key, very important keyword that I would make sure you write down. It does tend to pop up, and you should probably use it when you are talking about cells, particularly plant cells. So if it was moving in, definitely, but the water's moving out, so that's not going to happen. Okay. C, the sap vacuole would decrease in size. So the sap vacuole, which is seen here on the diagram there, the whole reason of the sap is to contain the excess water. All right, and that's what expands and gets smaller, and then that exerts pressure against the cell wall, which then gives it its turgid effect. If it's water moves out of it, then we would refer to that as the cell would be referred to as flaccid. Okay. So remember, we've got to go through all the options, but the sap vacuole would decrease in size. So far that sounds good because the water, remember, is moving out. D, the cell wall would act as a regulator of osmosis. Okay, what I need you to know about the cell wall is it is 100% permeable. All right, it regulates absolutely nothing because it lets everything in and everything else. Everything out. It is the cell membrane that does that job, not the cell wall. Okay. Some people do get that confused, especially when they're in pressure under the exam. So always take your time to go through the options. Okay. Question 10. Last of the multiple choice. So for thistle funnels, I think it's important that you circle that diagram and it shows you one. The table does show you that you have four, but just to make sure that you're really interpreting the question correctly. Each containing the same glucose solution. So whenever you see the word same, it's really important that it triggers a controlled variable. All right, we're placed in beakers containing different glucose solutions. So even if this question doesn't ask you about a fair test or anything, you should know that the glucose solution is a controlled variable and that the different each containing the same glucose solution in the thistle funnel were placed in beakers containing different glucose solutions. So therefore that's the independent variable. All right, so you can see colour in the thistle funnel. This would be the same, but then the solution on the outside within the beaker that it's placed into is an independent variable. This is the thing that's changed. Okay, and there should only ever be one thing that's changed. And in this case it is. The results after 10 minutes are set out in the table below. All right, take your time to have a look at what's in this table. So you've got the number of beakers. Remember, each beaker has a different solution down the bottom. The initial height. So this is exactly the same experiment pretty much that we did with the thistle funnels. So remember that you measured the height on that to begin with. All right, and you're going to use that to see whether the solution moves in or moves out, or should I say that the water will move in or out because the solute can't. All right, so let's go through it again. If the thistle funnel reading increases, that means that water has moved across. So it's moved in. And the only reason it would move in is because there was a higher concentration on the inside. Okay, because it wants to reach equilibrium. I'm just going to write concentration on inside. Okay, if it decreases, if the water decreases, water has moved out. All right, if water moves out, it means it's a high concentration. Water has moved out, which means that there's higher concentration on the outside. Okay, so we can see initial height, they all started at 3. So beaker 1, okay, beaker 1 has increased. So we can see that therefore that the type of solution that it was sitting, because remember that's the only difference. So if the height is increased, okay, that means that it must have been a lower concentration of solution than it was in the thistle funnel. With beaker 2, that's stayed the same. Okay, so I would say that that's an isotonic solution. So there would still be some net movement. You'd never say that there's no net movement. Water molecules would move back and forth, but the movement would be equal. Okay, beaker 3, it's decreased. Okay, if it's decreased, then that means that the water has moved out because there's a higher concentration on the outside. Because remember, the water is going to move to the highest concentration of the solute in order to reach equilibrium. It wants to be equal, okay? And then beaker 4, so the initial height again was 3, but then it's increased, okay? So keeping that all in mind, you might even want to just pause that and go through it yourself. Using the above information, it can be concluded that the glucose solution in 1, so just mark them, 1 and 4 were at a higher concentration than the glucose solution in the funnels. So remember, the water has increased. The watermark on the thistle funnel has moved up. So that means that there is a higher concentration on the inside, so in the thistle funnel, than there was in the solution that they were in. So let's reread that again. A glucose solution in beakers 1 and 2 were at a higher concentration than the glucose solution in the funnels. So if it was higher, the water would have moved out, but it's moved in. So we can get rid of A. Hopefully this is making sense. You might need to take the time to really write out a few beakers to see what's going on. B, the glucose solution in beaker 3, okay, so it's this one now, was hypertonic, okay? So remember what I said before? It shrinks, okay? So in this case, we'd see it as it moves out. All right, and if we saw that it moves out, okay, so R for shrinks means that it would move out. So that means, so the glucose solution in beaker 3 was hypertonic. The water moved out, which means it moved down, so there was a high concentration on the outside. That looks plausible. But remember, we need to keep going through the other options. C, the glucose solution in beaker 2, I'll try and change colour to help out here. Okay, beaker 2 could move freely between the funnel and the beaker. Well, remember we said that it was isotonic, but that doesn't mean that the glucose solution could move in and out. It just meant that the concentrations were the same, and therefore the water movement would have been the same. Okay, sorry about that. So that's not correct because the glucose solution couldn't move, it was only the water. All right. D, the glucose solution in beaker 4, so I'll change colour again. In beaker 4 was the solution with the highest concentration. Okay, so beaker 1 and beaker 4, they have both had increase. So we have a look. If the increased water has, because water has moved in, has higher concentration on the inside. So I would say that it's probably got the lowest concentration because the water is moving in. So instead of having the highest concentration, it would have the lowest concentration. Okay, so if we have a look back at the options now, the correct answer is B. I think that one is a little bit tricky. I would definitely take the time to go through and redo all the beakers so that you know what's going on. But make sure that before you do go through and have a look at the options, that you have done a sort of key with your results, so that you're not getting confused when you come here, because you need to start here first. Okay. How'd you go? Did you get 10 out of 10? 9 out of 10? 8 out of 10? Remember, you can always come back and do this topic test again, and see how you improve. I actually really encourage you to do that. So don't give up.